tightly affiliated with wetlands than, say, sandpipers are? And these are questions where a model can help nail down exactly what we mean by things like that and help put numbers on it. So that can be useful. The final thing that I think models are especially good at is scale. What I mean by that is, you can go anywhere into a bookstore and you can find a book that will tell you a lot about birds. The same thing with certain kinds of plants, other vertebrates, butterflies. But there are lots of taxa, most of them in fact, where we basically don't know anything. And the world experts know some, but they might not even know the names of all the species that they could be interested in studying, let alone enough natural history that they can put together a detailed picture of where they think the species will do well and where it'll do poorly. So I think models are useful for all those reasons. There's different ways you can evaluate a model, tell how well the model is performing at the tasks. Some of them are qualitative. Do you believe the results? Does the results seem to fit with some scientific theory? Do they help advance our understanding of this taxon? Others can be pretty quantitative. So here I've divided up those transects into two groups. The ones in white are the white circles are called training routes. So I show a modeling algorithm all the species that lived here, and then I tell it, take a guess. Look 150 kilometers away, guess what bird species you'll find. And if the model isn't very good at doing that task, if it can't make predictions, then maybe we don't believe what it says about species' responses to the environment or differences among species either. And for this talk, I'm going to be focusing on this data set of 368 species from the Breeding Bird Survey, split like this. And for this talk, when I talk about making predictions, that'll always be based on climate data that was collected from the WorldClim data set. And so the idea is that the model sees, oh, this is very wet and has a short dry season or something. Based on that, we can try to predict the species. Now, obviously, there are other things that are important for determining where a bird species can live, in particular land cover, and I'll get to that later. But for now, most of the research across most taxa, I think, in sort of a species distribution modeling framework is focused on climate, and that's where I'm going to start the talk as well. Okay, so that's sort of an introduction. Why do I think we need a model? What is the model supposed to do? But why do we need a new approach? Why can't you just run the same logistic regressions that people have been doing since the 90s? And I think logistic regression has a lot of nice properties that I'll talk about briefly, but it doesn't always perform as well as you'd like. So what logistic regression does, it's a generalized linear model. You start from the predictors, in this case, interaction between climate or between precipitation and temperature, and you try to get a probability that it'll observe a particular species. And we can represent the model as an arrow. The arrow just saying you start from here and you go to get a probability. If you want to make a prediction about another species, you fit a separate model, and if you want to make a prediction about a third species, you fit another model. And the really nice thing about this is that it's interpretable, right? So at the end, for each species, you get a coefficient that says how that species responds to each environmental factor in your data set. And if you want to say something like this species responds very strongly to warm climates and this species does better in cool climates, that's something you can read just right off the table. No matter how many species you have, that still remains tractable. The problem starts to come up when you start thinking from a scientific perspective, what is the process, or what's the community assembly process implied by something like this, when you've got separate models for each species and each one of them is generalized linear? Well, so it means that each species is basically independent of the others. No possibility of species interactions, no possibility that some unmeasured factor could cause changes in distribution or abundance. And so you're assuming these two things. Linearity, you're assuming that the shape of the species' response to the environment is known. And you're assuming that the species are independent of one another, that you can represent them well using separate models as opposed to one big one. I'm going to focus first, just briefly, on linearity, or in particular generalized linearity, because this is a generalized linear model. And what that means is that you're assuming that each species' probability of occurrence is going to have this shape in response to any environmental factor you measure. But we know as biologists there's other shapes that are equally plausible. Maybe suitability increases and then levels off, or maybe it increases at different rates. Maybe if you have a funny environmental variable, you could conceivably even have something with a intermediate minimum. And it turns out that people, especially in very high dimensions, are not yet at the point where, especially in taxa we haven't studied very well, we're not yet at the point where we can predict in advance, you know, oh, we're going to need this kind of shape as opposed to this kind of shape, as opposed to this kind of shape. And so what species distribution modeling over the last 10 years has done is it's traded our interpretability that we got from those generalized linear models, it's sacrificed that and in pursuit of accuracy. And it's let the machine try to figure out what the shape of those responses should be. And it does improve accuracy, but it means that we have a black box separating each species from the raw environmental data, which can make interpretability much harder. From a scientific perspective, the implied process that's associated with this set of models basically boils down to who cares, it's accurate. And we don't necessarily need to worry so much about making sure that what's going on in these black boxes, both individually and collectively across all the species, makes biological sense. And that can work really well, in fact. So I'm going to focus on an example called boosted regression trees. It's an algorithm for building these nonlinear models. It's the example I'll be focusing on in this talk, but there's lots of others. This one, the way the model is constructed for each species, each black box has 10,000 little models inside it that describe how the species' probability of occurrence relates to environmental factors. And so if you want to figure out where this kinglet is going to live, you need to look across all 50,000 parameters and all 10,000 submodels to figure it out. So that's what I mean by saying that there's a loss of interpretability. You can make plots like this that show response of probability of occurrence versus your environmental factors, but this is just a one-dimensional shadow of the full model, and it doesn't capture everything. So you could spend, a few hours or a few days looking at this from different angles and trying to figure out, okay, now I think I really understand the kinglet's response to the environment. But then you go to the next species and you have to start over from scratch because you've got 50,000 new parameters from 10,000 different submodels, and they're telling you a completely different story. So that's hard. So there's a big loss in interpretability, especially when you're looking across species. And I want something that scientists can use to help inform their understanding or corroborate or improve their understanding of species environmental tolerances and community assembly. And so this isn't really that suitable. So we've talked about how to generalize the linearity issue at the cost of interpretability. Next, let's talk about this independence issue that I referred to. Here, we've got completely separate models for each species, and so there's no way of sharing information across species about what kinds of environments they like, or if there's something that causes a correlation among species, there's no way that this model can talk to this model and say, well, I like forest and you don't, so we should be negatively correlated with each other. A little bit more concretely, we could have a situation where one of these black box models says there's a 30% chance of seeing a kinglet, another black box model says there's a 60% chance of seeing a lark, and then if we ask what's the probability of seeing both, it can't answer that question by itself. If we're willing to make the assumption that species are independent of one another after considering climate, as these two recent papers suggested, then you can put in numbers for this, but they won't necessarily be accurate. So in this case, these two species, as I alluded to earlier, have fairly different habitat requirements. And so the probability of seeing them both at the same site or along the same transect is much less than 18%. It's probably closer to 8%. So you can see these numbers shifting from independence to something that's a little bit more realistic. So that's not a huge change. It is a factor of 2 for some of these boxes, a smaller factor for some of the other boxes. But it starts to add up when you start thinking at the level of whole communities. So we've got 368 species in this data set, and the species richness might not be able to make as good predictions as you were hoping if you don't take correlations among species into account. So that's what these two papers I referred to earlier were about, and I'm going to show what happens if you use the methods they propose with independent models on the breeding bird survey data. So this Calabresi et al. Paper has a nice formula that you can use to calculate a probability distribution for how many species you would expect to see given a set of 368 black boxes. So right now, at this location, the model is the set of models is pretty confident that you'll see between, say, 60 and 80 species. At this other site, the set of models is pretty confident that you'll see between 30 and 40 species. And the thing to get from this is just that these confidence intervals are really, really narrow. Because the probability, you could think of each coin as an independent coin flip, the probability that only 20 of them are going to come up heads, or that 80 of them is going to come up heads independently, these are separate events, is really low. And we can plot, here's the predictions versus the observations, and here's the predictions that fall within the 95% confidence intervals, those narrow intervals. But the other half of the data set falls outside these confidence intervals. And so routinely, if you rely on the 95% confidence intervals from the independent set of models, you're going to be finding situations like this, where you were expecting to find 40 species, you built your MPA, or you built your park, or you built your nature reserve in this site, and it turns out that something isn't suitable, and there's many fewer species that can live there than you expected. One thing that I think is driving a lot of this, particularly at the large spatial scales with this data, is that there's more to ecology than climate. So I said initially, all of these predictions about how many species we'll find, or which species we'll find, are based purely on climate data. And it turns out that these sites were probably not depopulated species because of the climate was unsuitable. The climate-based models all say that you should be seeing 40 species. It's probably also not because these independent coin flips came up, oh, this species won't live here this time, but maybe next time. Oh, this species won't live here this time, maybe next time. Probably what happened is that there was some other environmental variable that reduced that systematically across species reduced the probability of occurrence. And that's something that you can't deal with if you have these separate models that don't talk to each other. Okay? So let's take a big step back, try to reconsider, how do we want to model this? In particular, how would an actual ecologist, someone like you guys, talk about community assembly? So they might say that there's a set of environmental conditions and resources. Maybe they'd use the word niche space to refer to this. There's a set of environmental conditions and resources, and if those conditions and resources are suitable for the species, they'll show up with high probability. And if they're unsuitable, they'll show up with low probability. So here's where the environment sits in niche space, and then if you will allow me to talk about niches for a moment, this is each of these arrows represents the species niche. It says which part of niche space can the species live in. So that seems like a pretty straightforward idea. The important thing to recognize, though, is that niche space, or the set of resources and conditions that species care about, are not necessarily the same things as the ones that we measured. Right? We can measure temperature. Maybe species care about some function of temperature. Or maybe they care about the interaction between temperature and precipitation. Or maybe they care about a whole bunch of other things that we don't even know to measure. Things like land cover, like that parking lot I showed. Or maybe they care about the history of the location. You know, it doesn't just have to be a forest. It has to have been a forest that hasn't burned in a certain amount of time. Or maybe there's other factors involving dispersal that affect which areas will tend to have a species of interest. Or maybe it's just something else that we ecologists with natural history understanding try to figure out what our species care about, but often we just won't know, or we won't know precisely. And so it would be nice if we could have a part of the model that represents the stuff that causes systematic changes in species composition, but we aren't necessarily sure what it is or how to measure it. We can represent that in a model by using random variables. So to instantiate this, we could say there's a distribution of possibilities. Maybe the environment is yellow, whatever that means, and then when the environment is yellow, this species has a higher probability of occurrence than expected based on climate, and these two species have a lower probability. Or maybe the environment is green and you get the opposite. So that's a way of introducing these kinds of systematic correlations among species into the model by having them respond to environmental factors even if we haven't measured it. So this is an acknowledgement that there are things that we can't know. In particular, climate is the only thing that matters. And we can talk about a distribution of possibilities rather than saying, this is the probability of observing the species. We can say, if the environment is suitable for the lark, then it'll have a very high probability of occurrence. If the environment is suitable for the kinglet, it'll have a high probability of occurrence. But probably you won't have a situation where both species will have a high probability of occurrence at the same site. Make sense? Great. So this is not a new idea. It's a moderately new idea, but it's something that has shown up from several different research groups in Europe and North America and Australia. And mostly they don't have this box here. They just send everything straight through. But mathematically it ends up being pretty much equivalent. And I'm going to focus in particular on one method that falls in this family called Bayes-Combs, which was written by Nick Golding. And he wrote this software as part of his PhD thesis in the UK and put it up online, and I discovered it and realized it didn't have some features that I needed, so I added them, and he made me a co-author. It's a really cool example of 21st century collaboration, especially given that we're on different continents have only met once. Anyway, so his software is pretty nice. It works reasonably fast with the big data set that I threw at it, with 300 plus species. And it gets this non-independence issue, as I was talking about earlier. The problem is, it's back to assuming that species have linear or simple responses to the environment. And so as a result, without these black boxes somewhere in the model, it doesn't do very well when we try to predict out a sample. And so it's capturing one aspect of the environment, species response to the environment that we'd like, but not all of it. And so my goal as a modeler is to try to be faithful to both the science and the data. And I think that these new models that have the random variables controlling co-occurrences among species are really nice. They have a interpretable process. It sounds sort of like what ecologists talk about when they talk about species composition and community assembly and so on. It doesn't assume species are independent of each other, which I think ecologists are on board with. You know, species are not existing in different universes where what happens to one has no information about what happens to the other. The problem is I need something, if I want to be accurate in my predictions, I need something that doesn't make this linearity assumption, which means that we're going to have to put some black boxes in, which is unfortunate. But we don't have to put the black boxes here, where we have completely separate models for each species' response to the environment. Instead, we could put just one box here. And so what that says is each species has a fairly simple, straightforward response to the environmental conditions in a given area, but that the relationship between the things that we measure and the things that species care about can be nonlinear and complicated. And I think that makes sense. It fits our biological intuition pretty well. And the other nice thing about it is that you can get coefficient table out of here. So you can look at this, you can look at this and see, here's how this species responds to niche axis 1, and it's the same niche axis 1 that this other species is responding to, maybe in the opposite direction. So that's nice, and I think it's much cleaner than this in a lot of ways. It means that each species you can describe their entire response to the environment with, say, a dozen numbers, instead of with 50,000 numbers. Like the 50,000 numbers you'd have here. Now you could put lots of different black boxes here. For boring computational reasons, I happen to put a neural network in here. And the model that I built that has this structure I called MISNet. And you can go and download it right now. The paper about it isn't quite submitted yet, but the software works, and it's not well documented, but it will be. I think this is something that people will really enjoy using in the future. So let me talk about, first, a little brief sidestep on how to fit this thing, and then we'll talk about what it's good for, and that'll cover the rest of the time that we have. How do you fit this thing? So I was trying to fit a model sort of like this, but more complicated, needlessly complicated, because I didn't think that something like this was possible. Then I found this paper by Tang and Salakudinov that came out last year in a machine learning conference. And they had an algorithm that lets you fit something like this called generalized expectation maximization, which was derived in this 1998 paper by Neil. What this does is it lets us iterate between two different steps. One that lets us learn about what the environment, these unmeasured things, what they must have been like. and one step that tells us how species respond to those. And it turns out that if you iterate between those two steps back and forth, you end up finding a local maximum likelihood estimate for the model, which I hadn't initially thought was possible. So in this step, it's pretty much standard species distribution models. You assume the environment is known, and you climb the likelihood gradient for each species. The algorithm for this is called backpropagation. The other step is that you assume that the coefficients are known, and you work backwards to what kind of environment must have produced that set of species with that set of coefficients. So here, it's like you're saying, wow, there are a lot of ducks. There must have been a wetland here. And so you get more information about these random variables. Then you go back and you say, okay, given that this place is a wetland, how can we improve our model's assessment of how species will respond to that? And then you find out, oh, it turns out sandpipers like wetlands too. And then you go back and you say, wow, this place must be even wetter than I thought. It has ducks and sandpipers. And then you go back and say, oh, it turns out, anyway. After you go back and forth enough times, you end up near a maximum, you end up eventually at a maximum likelihood estimate, local maximum likelihood estimate. So that's MISNet. That's what I've been working on for now. So let's talk about what this model can do. Hopefully to tell us, that, if not something brand new about birds, at least help quantify some things that we already knew, put them into a nice context. One obvious question is, well, what determines species composition? I gave the model climate data when it was making its predictions, and I didn't give it anything else. So unsurprisingly, the model says climate's really important for determining where species live. But it's not 100%, and it would be 100% if we didn't have those random variables describing things that we hadn't measured. Now, that's not necessarily saying that climate is the causal factor, but it means that you can take a species distribution like this and draw a box around it using climate variables and say that the species can be predicted well by climate. Other species might have some other causal variable. This species probably not responding directly to temperature, although it looks like, oh, it shows up in the north and at the top of mountains, maybe that is temperature. Probably it's responding to plants that are responding to temperature. The model doesn't know that, but it still shows up in that 71%, because I haven't told it anything about plants. But what about the other 29%? What about the stuff that can't be explained by climate, even indirectly? The nice thing about MISNet is that we have this coefficient table I alluded to before, and we can just compare species and see which axes of environmental variation show what kinds of patterns in terms of species affiliation with them. And also, because I'm working with birds, we can just go online and find out what kinds of things are important to most of the species in the data set, apart from a few naming inconsistencies. So I can combine the information from those two locations. We've got the classification according to the Cornell Lab of Ornithology's All About Birds website, and we've got the model coefficients. So the coefficients are going to determine the position in space, in this two-dimensional space, and the color is going to be based on the true classification according to the birders. And I'm just showing three of the classes here right now. The model, using a linear discriminant analysis on the coefficients, was able to do a very good job of separating species that were classified as living in grassland. They have very different coefficients, non-overlapping coefficients, with species that are classified as living in forest. And there's another axis of environmental variation that the model seems to be responding to that describes the same thing that the Cornell model, the Cornell birders, were talking about when they said the species lives in marshes or lakes or ponds. Now, obviously, there's a continuum between grassland and forest here, and if you plot species that are classified as living in scrub or in open woodland, then they show up in between. But what this shows is that the model was able to find a way of representing these axes of environmental variation that are really important for bird species composition without being told anything a priori about what the species care about, or even having estimates of how forested or how wet different areas were. All it had was climate, and it was still able to determine there's an important axis of environmental variation from species up here and species down here. And when we ask a birder, what do you think this axis of environmental variation is doing in the model, they can say, oh, that's a forest to grassland gradient. So that's a nice synergy between the modeling and the natural history. Now, some of you might be wondering, didn't we already know that water birds like water? Didn't we already know that warblers like forests? And yes, that's true to some extent, but remember, this lets us make quantitative predictions about where the species will be that might be more precise and more accurate than you could do just by looking at a field guide. Lets us quantify things about how much different species prefer to live in different habitats. And I think most importantly, it lets us scale up. So for most species, whether it's diatoms or bacteria, we're not going to have someone who helpfully colors in the different kinds of species for us. We're just going to have this blob. And but having a blob like this is still useful, because maybe we could say, well, this one is E. coli. Well, what do the other species near E. coli have in common? Or this species is something that causes diseases. What do the other species nearby have in common? And so I think that this is something that could actually help produce some interesting science in areas where we don't already know most of what's going on. Okay, but all that is only trustworthy if the model is accurate, if it's able to describe the community assembly process in a reasonable way. So to do that, like I said before, we're going to show the model these white points and ask it to predict a few hundred kilometers away what's at the blue points, what species are going to be found there. We can measure this in different ways. First way I'm going to measure it is species by species, what's the likelihood? So here I'm not giving MISNet any advantage because it can't use any information about correlations among species. And I'm going to use this boosted regression trees model that I described earlier as my baseline. So it's going to have a likelihood ratio of 1. Any model above this with a higher likelihood ratio is performing better. Any model below it with a lower likelihood ratio is performing worse. And Bayes-Com, the linear version of the model I showed before that had the unobserved environmental variation, doesn't do that well, according to this. So for the typical species, this means you're a few hundred times better off using the machine learning method than you are using the multivariate linear model. If you use a neural network, something that combines information across species and tries to form this idea of niche space that I alluded to earlier, you can do quite a bit better. So for the average species or the typical species, I'm doing about 20, 21 times better than you can do with these single species black boxes. So that's nice. But what if you start thinking about multiple species? This is something that I really motivated this work. So here I'm going to show, as you start adding more species to the community, the prediction problem becomes harder and harder, because if with one species you've only got 2 possibilities, presence or absence. With 2 species now you've got 4 possibilities, because they can both be present or both absent or one or the other, and it blows up really fast. And so the regression trees, their accuracy drops really fast from very close to 100% down to negligible as you start adding species. And MISNet shows a similar pattern, but the decay is much slower. So showing this another way, I'm going to show the gap in predictive accuracy, or in likelihood, between the 2 different models, and starting from the models being very similar, both getting most questions right, to by the time you're trying to predict a whole species assemblage, MISNet is tens of thousands of times better at predicting the community composition of a whole group of species, rather than trying to predict one at a time and assembling them after the fact. And it's a little bit hard to think about, what is a factor of 10,000 mean? In this case, I'm going to show with species richness. So remember, we were really concerned that the stack of separate models were telling us that we should expect an unreasonably narrow range. MISNet doesn't necessarily know, it has the same information, it just knows climate, and so it can't, the R-squared is slightly better, but not much. The big improvement, though, is that almost all of the points are now, that were observed, are now things that the model considers possible. Here, the model told you, no, you can totally rule out the possibility that there will only be 20 species, and you can feel safe thinking that there's going to be 40. MISNet is much more honest about what we don't know, and I think that's a big improvement. So that having these random variables in the model gives us a much more honest assessment of how much we know and how much we don't know about what happens when you have hundreds of species going on. So that's accuracy. I'm going to talk about one more cool thing that you can do with MISNet, and then I'm going to switch to talking about future directions. So a lot of us, when we go out and collect data, we have stray data points that either we didn't quite follow the protocol, or our car broke down halfway through the sample and we weren't able to finish it, or we have some other kind of information that's locked in a notebook somewhere, and we don't have a good way to incorporate it into our models, into our predictions. But it seems like it should still be useful. MISNet has a built-in way to do this automatically, which I'm really proud of. So here we've got a similar thing before. We don't know initially if the area is going to be grassy and suitable for the lark, or forested and suitable for the kinglet, but we know it's probably not going to be suitable for both or for neither. And if we average our predictions across all these different possibilities, we end up with something like this. Now let's say you had some external source of information that tells you there's a Nashville warbler that lives here. The Nashville warbler probably wouldn't have occurred there since it likes forest cover, and this guy doesn't even like tall grass. If we were up here, we probably wouldn't have seen the warbler, so we can downweight those points. If we were over here, it's very suitable for the kinglet, and it's also suitable for the warbler, so we can upweight those points. And suddenly our prediction shifts from thinking that with no information, we thought maybe the most likely or the best guess is here. We shift the entire set of predictions downward and to the right, because these possibilities are much more plausible, much more compatible with the observation of this warbler. And so it lets us update our predictions automatically. It can show here's the kinglet becoming more plausible in the presence, or once a warbler's presence is known, and here's the lark becoming less probable. But we can see there's a whole suite of forest species, and the model automatically learns, okay, these species are more likely to be present if their friend the warbler is here. And these species that all hate forests are much less likely to be present if the warbler is present. So I think that's pretty cool, and one way to think about this, an extreme way to think about it, but I think there's something to it, is we don't actually have to measure forest cover, because the warbler is able to do it for us. And in fact, one could even argue that the warbler is going to do a better job of measuring forest cover from a bird's perspective than we ever could. And so having the warbler act as an indicator of what the environment is like might be better in some cases, or at least a nice fallback option, compared to using GIS data or land cover data from satellites, which might not measure the same things that the birds care about. I have another example here. There's a redhead duck, which I did some research. Apparently these are really stupid even by duck standards. Hunters like them for that reason. And here's a similar graph. In the presence of a redhead, the model becomes more confident that you'll see this shoveler. This is a marsh breeding blackbird. And so on. There's a couple of species here that I thought were more interesting. These I tend to think of more as being grassland species than as being wetland species. But I looked, and this redhead actually does forest and grasslands quite a bit, and so maybe the model picked up on that, or maybe it made a mistake. It'd be interesting to talk with people who know more about birds and see what's going on there. But anyway, that's MISNet. That's what I've been working on the last few years. Has some nice features. It can automatically identify important environmental gradients and important environmental features, like this forest to grassland thing that I didn't measure. I didn't tell it what it was. But it discovered it based on who co-occurred. It does a more accurate job at predicting which species will live together. It builds more coherent species assemblages. It says if you have this warbler, you're also going to have the kinglet. If you have this duck, you're also going to have some sandpipers and rails and so on. It gives you this nice set of bioindicators for free. Once you fit the model, it's very straightforward to start adding in extra information after the fact. You don't need to fit a new model or build that into the model fitting process. You can just start with the model you already fit, and then every time you discover something new, you can use that to update your predictions very quickly. And I'm hoping that I've shown at least some potential that this can facilitate real scientific discovery. If not in birds, then in other taxa where we have less natural history information. Okay, so I'm going to spend the rest of the talk, the next 9 minutes, hopefully I'll finish before that, for talking about what's coming next. I've got 3 ideas in the pipeline that I'm pretty excited about. One is species interactions. So here, there's correlations among species, but as far as the model is concerned, what's driving them is a common response to the environment. And mechanistically, it would be interesting if we could include direct species interactions between pairs of species. Now, I think the one on the left is a black-capped chickadee, and this is a Carolina chickadee, but I forget. There are trained birders that have trouble telling them apart, so I feel a little bit better about that. And there's, according to the model I just showed you, these should co-occur a bit more than they do. One possibility is that they're competing. And I've built a model based on something called a Markov random field, a specific version of it called the Ising model. What it's able to do is put a coefficient here between the 2 species that indicates how much, when this species is present, downweight the probability that you'll see this species due to a direct pairwise interaction as opposed to this indirect thing through the environment that I showed before. And the same coefficient describes how much this species reduces the probability of seeing the first species. You can take these coefficients and then using either Markov chain Monte Carlo or a mean field approximation, you can figure out how often each combination of presences and absences should occur in the data. And in this case, I described, I'm getting too many of these, too many situations where both species co-occur. And so you can use that information to climb the likelihood surface and increase the negative association between these 2 species, make this line thicker, basically. And it turns out that works really well. I haven't incorporated it into MISNet, but I fit it in other models, one of which got rejected from PNAS, and it works. The problem, it starts to get harder when you start having more species. So with 2 species, you've got 1 connection between them. With 5 species, you've got 10 connections between them, and it blows up fast, but this is manageable. You just build into the model a prior expectation, which I think is biologically reasonable, that most pairs of species don't interact very strongly, especially not at the spatial scale of one of these 25-mile transects. You know, probably these 2 species, one chickadee is not going to competitively exclude another chickadee 25 miles away, and that's certainly going to be true for most pairs of species if you think about, like, well, are hummingbirds really going to be competing with ducks for much of anything? So we've got 2 causes of correlation in the model now. We can talk about it coming from these random variables or from the direct interactions. And let's not talk about that right now because I'm running out of time. I can talk about it in the question-answer period. So that's species interactions. Next thing I'm excited about is using species-level data, especially trait data and phylogenies. Because as I mentioned at the beginning, we know a lot about birds, and it seems like we should be able to use that either to test hypotheses or to inform our models. So for instance, we might hypothesize that similar species have similar coefficients governing their response to the environment. Whether that's similar in terms of traits or similar in terms of phylogeny, we might hypothesize that species that have similar roles in the environment or similar evolutionary history will tend to have similar responses to the environment. We might also hypothesize that they're more likely to compete with one another. And I think both of these are interesting hypotheses that you could test with a model like this. And there's 2 ways that you could go about this. One, you could fit a standard MISNet model like I showed, and you can test for the effects of similarity. So for example, I did a Mantell test asking, do closely related species have similar coefficients? And the answer seems to be yes, although I haven't done this carefully enough yet. And it seems to be driven by certain taxa, like the waterfowl, that there's a consistent environmental signature of being in that taxon. The other option, and one that I think is more interesting, is you can try to build in that assumption that similar species will have similar coefficients directly into the model. And that would allow you to say, well, I don't have very many observations of this species, but I know what family it's in. And I know what other members of that family, how they respond to the environment. And that gives you some baseline information. So I'm pretty excited about incorporating species-level data for that. And I think plants would be another great taxon to work with, because there's good trait data there. Finally, I talked a little bit about these bioindicators, where you have some extraneous data that you aren't sure what to do with, but you can use it to improve your predictions. I want to go much bigger scale with that. So I've talked a little bit about doing that within the breeding bird survey data set. Breeding bird survey is wonderful, in particular the data is fairly high quality, the detection rates are pretty good for the most part, but it's dwarfed by eBird, which gets about 1 breeding bird survey's worth of data a week or every few weeks, depending on how you count. And it would be nice if we could combine these sources of information in some way. You know, using the data which on average I think is more reliable from breeding bird survey, combined with the fact that eBirders are everywhere. You know, I showed like 2,000 points up there that showed the breeding bird survey. Again, eBirders are everywhere, and so you can get much more information. One interesting way to do this, I think, would be fit the standard NISTnet model like I showed before, but then use eBird to guide the model towards, well, it looks like there might be some waterfowl in this area. Maybe you should update your predictions in that area, in that direction. And so you can hopefully do that purely based on these observations, which came from a different sampling method, a different group of observers, different skill levels, and so on. And hopefully this wouldn't run into some of the same problems you would find if you tried to analyze the eBird data directly, just because it's so heterogeneous. And like, I reported some eBird sightings. I don't know why they allowed that, but I didn't report everything I saw. But you can be pretty sure that when I say that I saw a western scrub jay, I saw that. And maybe that's a little bit of useful information that could feed into a model like this. It also doesn't have to be restricted to birds. There's lots of other data sets, and I've just shown a couple of projects here. We heard about iSpot earlier that could benefit from this, and going in both directions, I think. So herps and mammals, I think, often are harder to sample than birds. Sometimes you need special equipment, like traps, to sample mammals. Sometimes people are less well trained in terms of finding herp calls. But it would be fantastic, I think, if, like, rather than relying solely on data about salamanders to tell you where the salamanders are, if you could use information about other taxa, like birds, where we have a lot of data, you could say, well, there were some rails and sandpipers here, so maybe there will be salamanders here too. I think that's an interesting idea that could be like a force magnifier, right? So like, you go out and you put out your pitfall traps in some areas to find the salamanders, but this gives you some information about areas you can't sample directly. So let's just wrap up. I said all that already. Okay, thank you. And feel free to go. Answer any questions.
So actually, before you do that, I just want to make a real quick announcement. So I know some people may have to run out because it's 1 o'clock. That's fine. There was pizza ordered, so anybody who wants to hang out here and eat pizza with the candidate and discuss the research further, grad students, I mean, undergrads, I guess, you can do that. It's not going to be necessarily running up or lunch.
Are there e-birders in Nevada?
Probably. Yeah. Or you had
So
yeah.
some of these data sets give you abundance data. Can you modify your models to not only get presence, absence in the community, but relative abundance?
Yeah, absolutely. So the question was about getting abundance data, and the answer is yes, you could. I think for the species interaction part of it, for technical reasons, getting the direct pairwise stuff between species is really hard with abundance. But for the presence, absence stuff, but for the rest of the model, absolutely. I think abundance is really interesting. I wonder sometimes about like what I should do with some of the abundance data, because like I'm imagining a breeding bird survey person going out there, they have 3 minutes to count all the birds in the area, and they're like, okay, here's one duck species, and there's this sandpiper, and then suddenly like a flock of starlings comes by, and they've got to write down, okay, that's 987 starlings, and then go back. So I'm not sure what the best way to do it, but there's nothing in the math or the computation that would prevent me from doing it. And I think that potentially is a really valuable source of information. Glad you brought that up. Yeah?
What about temporal component? So you've got breeding bird survey, are you using 1 year, are you aggregating across years? Can you build like a time series associated with that?
So I did everything within one breeding season with eBird, with breeding bird survey. But I think it would be really interesting to make the model more sensitive to changes over time, either within breeding seasons or across years. Or, you know, the eBird data is constant, you know, probably less in the winter, but still definitely have people collecting the data. And they have these beautiful maps that show density of birds migrating in and out of North America every year that really recommend people take a look at. And so thinking about the time series, it adds a lot of difficulty in some ways, particularly for species that migrate, but I think it could be really valuable. Yeah?
Well, actually, I was thinking about time as well, Sean, but I was also thinking actually a much deeper time series. So have you tried playing around with, creating these models for current time periods and hindcasting? Because obviously that's the biggest question with the paleoecologists is the
Yeah,
turnovers.
yeah. So I think the hindcasting is a really interesting question. One thing that I think this model could be especially good for in that context is, well, here. Yeah. So here, I was talking about using the species you observe to inform us about what the environment must have been like. And from what I understand, that's something that paleoecologists are really interested in doing. And I think that a model like this might have certain advantages over some of the alternatives that I've seen in the paleo literature. I think it would be a really interesting comparison, or maybe we could both learn from one another, with how do you learn about the past from just these fossils that you see. I think more concretely, thinking about going back to 1960s when the breeding bird survey started could also be really interesting. Is there, yeah, either.
Oh, okay. You mentioned that you might use warblers in some sort of an optimality assessment of like forest cover, where you would think they're better at, at estimating forest cover than
or
we
not
are.
at estimating forest cover, at estimating forest quality from the bird's perspective.
Yeah,
Right.
Yeah. But what happens if the birds are lousy at it?
Well,
what happens if they're way suboptimal?
Well, so that's an interesting question. So here I'm basically
it could be
assessing,
restricted to a habitat and still be bad.
Right. So you're right. There's all sorts of subtle distinctions that I'm glossing over with something like this. The distinction between presence and positive fitness in an area is one of them. The distinction between not observing something and it not being there is another distinction. So when I'm being careful, I like to think of this as not necessarily making predictions about the true underlying process, which we can't necessarily get at directly in most cases, but it's making predictions about if we had another breeding bird survey person go out with comparable skill to the skill of the people that were here, what would they find? And the deeper questions about like how many eggs are being laid and how much nest predation is there are things that are hard to get at with observational data. I think they'd be hard to get at with experiments too. But yeah, that's absolutely something that it's important to keep in mind. Yeah.
So I was wondering if you had redone the analyses just using the eBird data rather than just the bird data. Because essentially, other than birds, if you go to any other taxa, you don't have a breeding survey of that same quality on such a large scale. So I'm just, given if you have much crappier data to begin with, how good can, what can you expect out of it?
Right. Well, so as a modeler, I get to pick what systems I work in. And so I've chosen the breeding bird survey because it works well for my needs. I have worked with eBird for another project, and my experience so far is that the variation among observers and other sources of variation, including variation in sampling methods and effort, is so big that it can be hard to pull out a signal. And it's also it's so big it's hard to download. You can't keep the whole thing in RAM. It's someone who's a better computer scientist than me, or better data manager than me would have much better time with that, I think. But yeah, I think eBird, it is what it is, but it's really wonderful. Just that.
Right. Or even GBIF. So something that exists for more.
Well,
for more
how much of
taxa.
GBIF is coming from eBird? It's like 2/3 or something, right? More.
Yeah. Well,
so
for things that don't have as much observation.
Anyway, yeah. Anything else? Yeah.
So this is a tough question, but I kind of feel like you have the pieces, but I wonder if you can do it. So one of the things that I've always been interested in, if you look at any group of taxa that you're interested in, they are present in certain habitats and they're missing from other habitats, from other parts of their distribution.
Right.
So the question becomes, could you build a model or a Dept part of your model so that you could co-estimate the probability that the environment is suitable and the taxon is based on presences, and that it's suitable but it's not there, which may be related to some interaction term? Because it seems like you could measure the interaction terms and the suitability. So you can make a statement saying, this bird should be there, but it's not there because of these other taxa are there.
Right.
And
So
test that.
right. So my model makes some distinctions between why species don't occur. So, you know, I can distinguish between something correlated with climate and something not correlated with climate. And among the things that are not correlated with climate, I can look at it some by looking at the other species in the area and see, like, maybe if no one is there, then I suspect someone paved it over. If nobody in this clade or in this guild is present, then maybe it's because the habitat wasn't suitable. There's all sorts of other things that could happen though. Like, people have talked about the possibility that there's systematic biases where older birders can't hear the highest pitch bird calls, and then that, you know, so I haven't looked because I don't have the, I don't know where to get the data for this, but it would be fun to see, like, if one of, so I plotted that scatter plot that showed, like, here's the response to what I'm calling forest cover, and here's the response to what I'm calling wetland cover. Is there another axis of environmental variation that NISNA is picking up on that corresponds to the pitch of the call? Or things like time of day that they're out, or things like that. I think there's an indefinite number of things that could be driving deviations from model expectations. And the nice thing about having lots of species in the same model is you can start to narrow it down, right? Because you can triangulate and say, well, if it was this factor, then it would have affected this species too, and things like that. Yeah.
Just adding on to that, how about putting humans in?
Yeah,
I
they're
mean, a huge variable, right?
yeah. So I allude to that when I talk about parking lots, but yeah, I think there's good human data that people use. So I know some of the eBirders have used population density, land cover, including things like impervious cover, parking lots and buildings and stuff. There's also, someone suggested, and I think this would be interesting, using maps of basically light pollution. You know, you have a satellite up and you look down and see what areas are bright at night. Could that be an interesting factor, either as a correlate for population density or land use, or as something in itself? I think, you know, some of these data sets are easier to work with than others, but I think it could be really interesting. You know, there's the sky's the limit for this stuff. I've got some techniques that I think are pretty good, and part of the thing is just finding good data to throw at it. Because the better the data, the better the predictions should be. The better, more insight, hopefully, we can get into what's going on.
So I think on that note, we'll call the questions to an end.
Okay.
And thank you again for an excellent seminar. The pizza is here, I'm told.
Jeremy did this. You're trying to compete with John Travolta.
I don't understand